In this video, we will have a brief overview of the structure and function of the respiratory system. Our respiratory system is made of the airways and the lungs. The primary function of the respiratory system is gas exchange. This is where our body brings in oxygen needed for aerobic metabolism and gets rid of carbon dioxide, which is the byproduct of metabolism. Other functions include inactivation of vasoactive substances such as bradykinin, and the lungs is where angiotensin-converting enzyme, or ACE, is found. This enzyme converts angiotensin 1 to angiotensin 2 in the renin-angiotensin-aldosterone system. Air enters our respiratory system through the larynx in the trachea, which then branches into the bronchi. Our bronchi are lined by specialized cells that you can see in this box that are called pseudostratified ciliated columnar epithelial cells. The bronchi also have mucus-producing goblet cells. The hair-like projections on our epithelia, called cilia, and our mucus create the mucociliary blanket. This is an important protective factor that allows us to capture unwanted particles in the airway, such as pollen or microbes, and move it out of the airway so it doesn't cause infection. Our bronchi branch off into secondary bronchi, tertiary bronchi, and eventually into bronchioles. Gas exchange takes place in the terminal respiratory bronchioles, specifically in the alveolar sacs and alveolar ducts. The alveoli are surrounded by a capillary system, and together they make the alveolo-capillary membrane. This is where gas exchange occurs in the lungs. In our alveoli, we have several types of cells. We have type 1 alveolar cells, which are large cells and provide most of the surface area within the alveoli. These are the cells that provide the barrier between the air and the alveolar wall. Type 2 alveolar cells produce surfactant. Surfactant is a substance that decreases surface tension, making inflation easier. Without surfactant, the lungs or the alveoli would stick together, much like a balloon that you have blown up and let the air out, where it then sticks together and is harder to pull apart. We also have resident immune cells, macrophages, within the airways to help us monitor for and destroy unwanted substances and microbes. Our lungs are housed within the pleural cavity. The pleural cavity is a thin and transparent double-layered serous membrane. You can see in this picture where it lines the thoracic cavity and completely encases the lungs. There are two layers to the pleural cavity. Inside here you can see the visceral pleura, and outside there is the parietal pleura. These pleura are separated by a pleural fluid. The movement of air into and out of the lungs is called ventilation. Ventilation is driven by different pressures. The main pressures to think about are the airway pressure, which is our atmospheric pressure. This is the pressure in the air outside you, and that's the same air we breathe in. At sea level, atmospheric or air pressure is 760 millimeters of mercury. The pressure within the alveoli, or the intraalveolar pressure, will vary throughout the breathing cycle, and as we move through this, it will make more sense. Another important pressure is the intrapleural pressure. This is the pressure within the pleural membrane. This pressure is called negative pressure because it's lower than atmospheric pressure. It is only 756 millimeters of mercury, so 4 millimeters of mercury lower. This intrapleural pressure is negative, and it provides an important function because this negative pressure provides a pulling pressure, a pulling force that keeps our lungs expanded. Without that pulling force, our lungs would collapse. For example, if someone had a puncture to their lung, let's say right here, allowing the lung to be exposed to atmospheric pressure, that lung would collapse because it doesn't have that negative pressure pulling those sacs open. Again, sometimes it's easier to think of the lungs as a balloon or even a water balloon that you need to pull to get it open. Let's take a little side trip and think about some pressure situations. So Boyle's law describes a relationship between volume and pressure in gas. Basically, Boyle's law says in a container with gas, as the volume increases, the pressure decreases. And if you increase the pressure, if you decrease the volume, that pressure will increase. If you were to connect these two containers by a tube that have different volumes and different pressures, gases will move from the higher pressure, or the lower volume, to the lower pressure, to the higher volume. So just like other substances that move from high to low concentration, our gases are going to move from high to low pressure. This is important because ventilation is driven by differences in pressure. The air we breathe outside is at atmospheric pressure. When we breathe in, our lungs will expand, and then our thoracic cavity gets bigger. That's going to decrease our intraalveolar pressure. At this point, atmospheric pressure is higher than intraalveolar pressure because we've increased the volume of our lungs. That allows air to flow into the lungs. So before our alveoli were smaller, we breathed in, making it a larger container, and air is now, which is gases, right, is now going to move into our alveoli. When we breathe out, our lungs recoil or get smaller, and so that's going to decrease our lung volume. Our intraalveolar pressure is now higher than atmospheric air. And so because the intraalveolar pressure is higher than atmospheric pressure, where we've made the container smaller in our alveoli, that is going to push the air out of our lungs. So to recap, when we breathe in, our lungs expand. That lowers the pressure, allowing air to move into our lungs. When we breathe out, our lungs get smaller, it contracts. We now have a smaller volume, so the air is going to move out of our lungs back into the air. Let's look at another gas law. It's important to understand that when gas molecules are in contact with a surface, they exert a pressure. Just because they're a gas doesn't mean they don't exist, right? So they're going to have a pressure. Well, Dalton's law said that each gas in a mixture, so here we're thinking about air, exerts its own pressure. So our air is made up of oxygen, nitrogen, argon, water, carbon dioxide, and many other substances. And they all come together to make air. So each one of these gases is going to make its own pressure: oxygen, nitrogen, carbon dioxide. And so we know that gas will move from a higher to a lower pressure, and that includes partial pressure. So if the air pressures are equal, but one container has a lower partial pressure, then that gas will also move. And then Henry came in and added a different piece to that. So we know that gases can move into liquids, and so the partial pressure of gas is going to drive that. But there is one more factor, and the solubility of the gas also determines the concentration of a gas in a liquid. So for example, we have a lot of nitrogen in the air, but it's not very soluble in blood. So outside of high pressure scuba diving type conditions, nitrogen is not normally going to move into the blood in significant volumes. Now we'll talk about ventilation and perfusion. So we mentioned ventilation, and that is movement of air in and out of the lungs. But in order for us to exchange this gas, we must have perfusion. Perfusion is the flow of blood in the pulmonary capillaries. You need to have the air and the capillaries be at the same place for gas exchange to occur. And so that means we need equal ventilation, air moving in and out of the lungs, and perfusion, flow of blood. An important concept to understand related to ventilation perfusion is that if these equal amounts aren't present, then you'll have a ventilation perfusion mismatch. That means either ventilation or perfusion is not equal to where it should be. This ventilation perfusion mismatch is the most common cause of what we call hypoxemia, or low oxygen in the blood. So we'll take a brief look at what a ventilation perfusion mismatch is. So remember, ventilation is the movement of air into the lungs. So I've put that in blue because our air is going to be blue. And perfusion is the blood flow through the pulmonary capillaries. And for some reason, that is a Q. Probably because perfusion clearly starts with Q. I put this in red because it's our blood flow. Movement of air into the lung, ventilation, should equal blood flow through the pulmonary capillaries, or perfusion. But you could have two types of mismatch. It can be a high mismatch or a low mismatch. A low mismatch, we call it a low VQ. Here, as you can see in blue, the problem is the V. You have perfusion, but there's no ventilation. This is also called a shunt. In a low VQ, something's wrong with the lungs. That is handy because low and lung both start with an L. Some examples that you'll learn about that are a low VQ mismatch or a shunt are atelectasis, where you have a collapse of the alveoli, or pneumonia. You can also have a high VQ. This is also called dead airspace. You see I've highlighted the Q in red. That means you have ventilation, but you don't have perfusion. So something is wrong with the blood flow. An example of this is a pulmonary embolism. It's important to understand the difference between a low VQ and a high VQ. What could cause them, and what the effects could be. This is the end of our respiratory system video.